Welcome to Shaping Science, a collection of videos from the voices that are shaping science today. I'm Nikki Spahich from The Scientist. Today we're highlighting the groundbreaking technologies that won our 2025 Top Innovations contest. After perusing numerous nominations for cutting-edge products, an independent panel of judges selected the most exciting technologies for scientists from the laboratory to the clinic. Here are the winners in the lab research tools and emerging startup technologies categories.
meCUT&RUN is an extremely efficient and cost-effective method to map DNA methylation locations genome-wide. CUT&RUN stands for cleavage under targets and release using nuclease. So what's novel about meCUT&RUN is that it takes advantage of a naturally occurring 5-methylcytosine reader protein called meCP2, and it uses this as a molecular probe. What led us to developing this technology is finding a cost-effective solution to mapping DNA methylation genome-wide because existing enrichment falls short in a variety of ways. There's a chasm between whole genome bisulfite or enzymatic methylation profiling and enrichment-based approaches. Whole genome captures every single base pair location of DNA methylation, but this comes at a massive sequencing cost, whereas enrichment assays achieve to provide a cost-effective way, but they all fall short in terms of how much of the genome or CPGs they capture. And so our goal was to close this gap with meCUT&RUN without being limited to a small fraction of the genome. meCUT&RUN examines the entire genome using meCP2 as a probe. And notably, meCUT&RUN captures 80% of all methylcytosine at 20-fold reduced sequencing costs using as little as 10,000 cells, so it's really sensitive to input. And it avoids the bisulfite conversion, which minimizes bias, and optionally, enzymatic methylation can be performed for base resolution profiling.
In all of single-cell sequencing research, one thing that's been pretty interesting is year over year, the coverage experiment that's being done is growing at essentially an exponential pace. Folks are actually looking at doing, many millions of cells for a lot of studies. And so the question becomes, how do you scale with that, with the needs of kind of the scientists and the different questions that they're asking and what they actually need to do to answer their biological questions. And so that's really been the whole drive for launching the WT Penta, which is the most scalable single-cell RNA sequencing platform available, and it can do now 5 million cells in one shot. That can also scale up to 284 samples. What we're doing is actually capturing the whole transcriptome within single cells. We can start to understand what different cell types exist within what could be a very complex biological system. Once you've done that, you can go deeper and ask, okay, what are the biological mechanisms within those cell types and which genes are being turned on and off? Cells and samples are kind of the two key axes. So the number of cells you can profile is, again, largest you can possibly do. And that's really, really powerful for people who are doing, say, high throughput like drug screens and CRISPR screens, where the number of cells directly translates to the resolution of their data set. And there's other areas like population profiling. So large population consortia are now, in addition to whole genome sequencing, might want to also be getting now single cell RNA-seq data from those samples that they're collecting. And the question is, how do you actually profile hundreds of thousands of samples? And so how do you really do this in a scalable way? And that's really where multiplexing large numbers of samples at a time is really, really exciting to them.
The sponge is basically a lyophilized, so a very dried alginate complex matrix. It's full of little micropores, and it's used for activating and transducing T cells. So you basically put T cells and virus onto this material, and they absorb through all the little micropores. And that causes the virus to come into very close contact with the T cells and basically allows the virus to very, very effectively enter the cells to try and get T cells to take up viral constructs. It's not easy. It's actually fairly a big challenge. So you have to purify T cells. There's like multiple rounds of purification with beads to pull down T cells specifically. And that's a lot of hands-on time and effort. You then need to activate the cells. You need to treat them with certain antibodies and cytokines. And again, you do that in the context of beads, which you then need to remove. And then finally, you need to actually put the virus on them and transduce them. It's actually a fairly complicated process. It's typically using hours of centrifugation. So all these are pain points. We wanted to think of a way of making that simple. And the sponge really does that, because essentially you can go directly from PBMCs. You can put those directly onto the sponge because it's specific for activating T cells. And at the same time, you add the virus, and the micropores force the T cells and the virus into close proximity, and that induces them. So all in that one very simple step just makes the whole thing very simple.
Vega for us is an exciting innovation in terms of it really is the first benchtop hi-fi based sequencer. It brings our well-known, highly accurate, long-read hi-fi technology to a whole new set of customers and enables them to access it in a way that they never could before. So, we've really invested deeply in making this the most affordable, easiest to use, most accessible long-read sequencer that provides high accuracy data that the market has ever seen.
Bringing that innovation that David talked about was really important for these mid-sized labs, some core facilities, some smaller emerging biotech companies, people that want the accuracy without all the infrastructure requirements. So really excited by the way this is taking off. It is helping us in microbial genomics. It's helping us with targeted human disease.
This is designed to be the most accessible sequencer we've ever created. There was so much demand and excitement for the hi-fi data type that we produce. We've really tried to minimize all the complexity in the system. So the only things you need to do when you get a Vega is get it on the bench, plug in the power cord and plug in a network cable, and then the instrument is pretty much ready to go.
If we're going to make it more accessible, then you do have to make it very simple. And so we took all of the same workflows and automation and intuition we had from the higher throughput platform and built that into the Vega from the beginning. And I really do see this as a gateway to start to get more people to be able to access hi-fi and start to build out the understanding of disease and biology with these tools being more available to them.
What I've been developing over the last 25 years is a way of making molecules where we can control their three-dimensional shape better than pretty much anything else that's out there outside of biology. Nature, when it makes proteins, it takes these amino acids and it links them together through a single amide bond, and there's rotation between every pair of amino acids. And so you get these long chains that have to fold into a three-dimensional shape to do the magic that they do. That folding process is very difficult to predict. To design molecules that way, it's just really roundabout, and I found it to be very frustrating and difficult. So I thought, how do we get rid of that process? Why don't we make building blocks that we link not through one bond, but through two? So we get a ring, and then we make the building blocks rings, and then we have all these fused rings. Making fused ring systems takes an enormous amount of work, years to make micrograms. It's just not scalable. We came up with a scalable way to make molecules that are fused rings. They're derived from an amino acid called hydroxyproline. We do a lot of work on it to turn it into a building block that can couple to two other building blocks through a ring. We can make those building blocks on multi-kilogram scale and absolutely pure. And now they are like molecular Lego, and we can snap them together in any order. We're going to develop molecules that can bind proteins the way that antibodies can, but have better drug properties. Molecules that can get into cells that you can take as a pill, just making molecules that are targeted towards their target protein and only bind that. So safer, more selective drugs that have better drug properties than peptides, antibodies, and even small molecules. That's the goal.
So the Vega is the world's fastest live cell imaging system. So it essentially combines 24 microscopes into one compact benchtop system. And that enables us to deliver unprecedented scale with regards to high throughput screening, as well as live functional drug studies or interaction studies among cells and organelles. It works really well for capturing large samples or mini samples at once. So if you imagine a 96 well plate, for example, we can very quickly scan an entire well plate in under a minute. We also can do analysis integrated in, so we can complete the whole imaging acquisition and analysis process in anywhere from seconds to minutes, depending on the number of parameters you have, number of fluorescent channels, number of stats, etc. We're really trying to take the process of image acquisition for many samples at once from hours on other systems down to minutes and even seconds. That will really save a lot of time from the lab, just having to go through such a manual process, stepping and scanning well by well with traditional microscopes that tend to have just one objective as compared to our 24. And then because we have 24 objectives, we can allow video recording for functional studies. We can truly capture live video of 24 samples at once. So the main challenges that we're addressing are really throughput and also just live imaging at scale.
PCR technology has been around for 30, 40 years. And you see actually every 10 or 15 years, there is a leap in innovation. When you look into these technologies, they're actually all just estimating quantification. What Countable PCR is actually a true quantification, like DNA counter in the PCR format. So if you have 100 molecules or you're looking at 100 viruses, for example, in a sample, we can give you that number that you have exactly 100. And the reality is that our technology delivers true quantification, no sample loss, and you get like basically single molecule resolution data for genomics. We can separate a simple PCR reaction into 30 million individual picoliter reactions. Now you start separating the targets you want to see in the individual digitized signal, and then now you can actually count them. So the magic of the sauce, right, is really the ability to divide a reaction into 30 million compartments. When you are able to divide a reaction into 30 million compartments, whether you're looking at different targets and whatnot, because they're all isolated in single molecule compartments, turns out multiplexing or developing new assays can be a breeze. A lot of times, within a week, we can develop a high quality assay. When you can actually divide a PCR reaction that crazily, things change. Higher sensitivity, higher quality results, true quantitation, and assays get developed quicker.
Congratulations to all of our winners. To learn more about each innovation, click on the link below. And stay tuned for our 2026 competition, coming soon. Thank you for watching. Together, we are shaping science.